¿Comenzamos?
1 minute, please. Yeah, no, it's okay.
Jorge, voy a dar umas palabritas primero para los alumnos. Bom, boa tarde todo mundo. Hoje temos o seminário do professor Jorge Casal e, como sempre fazemos, podem colocar no chat do YouTube ou do Meet as perguntas, mas Jorge também concorda e gosta que, se for o caso, interrompam com perguntas para não se perder na palestra e peçam esclarecimentos quando necessário, tá? Então, fiquem à vontade, por favor, para participar e tirar dúvidas com ele em qualquer momento. I will switch to English. So, hello everybody, good afternoon, and it's a pleasure for me to present today our speaker, Jorge Casal. He's an internationally recognized researcher in photobiology. And Jorge, thank you very much for accepting our invitation. And I'll pass to Leandro for a little introduction about your CV.
Okay, thank you, Professor Magda. Thank you again, Professor Jorge, to participating. So, Professor Jorge Casal undertook his early studies at the University of Buenos Aires, where he completed his Master's Science before moving to the University of Leicester, UK, where he completed his PhD. He is now professor at the Faculty of Agronomy of the UBA, a senior research investigator of the National Research Council of Argentina, and at the Instituto de Investigações Fisiológicas e Ecológicas Vinculadas à Agricultura, and the head of lab at the Instituto Leloir, Buenos Aires, Argentina. His research interests include the signaling network involved in plant responses to light and temperature cues and its functional implications in crops. Professor, thank you again. The class is yours.
Okay, thank you, Magdalena. Thank you, Leandro. Today we are going to talk about the environmental regulation of hormone signaling in plant growth plasticity. So the first couple of slides are going to be...
Jorge, excuse me, just could you put in the presentation mode?
I have it in the presentation mode.
Do you have two screens? Are you using two screens?
No, just one, as far as I understand. Okay.
For us, it's not in the presentation mode, right?
Okay. I will try to share again. I will stop sharing for a moment.
Okay.
Came out of here. Go back to...
I think it's better to select the sharing all screen, not only window.
There?
Not yet, just the PowerPoint, but not the presentation.
Okay, maybe the whole screen then.
Yeah. Okay, we can see your screen.
Yeah. Is it okay now?
Perfect.
Okay. Okay, then we are going to talk about environmental regulation of hormone signaling in plants, how that affects growth plasticity. So the first couple of slides, as I mentioned before, are going to be related to growth plasticity, the meaning of that. Well, plants have to live in a rather complex environment because the environment, in a way, is changing all the time. For instance, here you can see a soybean canopy, soybean crop from above, and a soybean crop from beneath. And the amount of radiation received by a given organ of the plant will be changing, for instance, in a couple of seconds with the movement of leaves with the wind, which project or do not project shade. It's going to change throughout the day with hours throughout the day as solar elevation changes and sunlight penetrates some regions of the canopy and not others. And it's going to change in the matter of days or weeks as the crop grows and the plants become more heavily shaded each other. It is also going to change in terms of months, because at different seasons the radiation load is going to be different. So light changes in a wide range of time scales, and so is temperature. And then the plants can be in a soil with more or less nitrogen and other nutrients, and there can be more or less water, and all these variables are changing to some extent independently. So you can have a shade with or without water, shade with a high temperature or a low temperature, and so on and so forth. So the environment is complex and it changes with time. So there's not an optimum plant body to fulfill the optimum function, because there is no one environment. There is a combination of environments that the plant is going to experience through its life, and the only way to be close to optimize under different conditions is to change when the conditions change. In our laboratory, we concentrate on the analysis of plant responses to shade, and during the last years we have also started to work on the plant responses to temperature, because there are some similarities between these two responses, which I think we are going to understand in the next couple of slides. This is, for instance, a typical response of an Arabidopsis plant to shade. On the left, we have a plant grown at the sunlight, and you can see there that the embryonic stem or hypocotyl is quite short, whereas at the right we can see a plant grown under shade, and the hypocotyl is quite long. The plants are much taller, and if we think of these as plants grown within a canopy, this plant has a larger chance to impact more light, because deeper within the canopy, the shade is more intense. So this increase in the growth of a hypocotyl helps the plants to avoid shade. That's the reason why these are so-called shade avoidance responses. And this is one example of plasticity, because the plant is going to do better under shade with this shape, at least individual plants. It's going to do better with this shape under shade because it has a better opportunity to get light. But it's going to be better with these shorter hypocotyl in places where there is a strong sunlight, because this is going to receive, for instance, less impact from the wind. Of course, this is apparently a rather simple plant response in a simple plant such as Arabidopsis thaliana, but similar things occur in crops. And let me show you just one example. The rest of the talk is going to be with Arabidopsis thaliana, but these are crops of sunflower. At the left, we can see by the end of the season, plants grown at a density which is very close to the commercial density in Argentina, about 5 plants per square meter. And at the right, we have a much higher density, 14 plants per square meter. We can see that at the left, the plants the stems have adopted a more or less straight position, whereas at the right, the stems are inclined. And if we look at this in further detail, we can see that one stem goes to the left, the next goes to the right, left, right, left, right, left, right, left, right, and so on, with very few failures, which this is actually a wave that propagates through the row. And there are these apparent mistakes only at the places where different waves originated at different places of the row encounter each other. So why is this self-organization of the crop, how is that produced? Well, if for any reason a plant becomes slightly shaded by its neighbor, because for instance it's slightly smaller, it bends in the direction opposite to the neighbor in the search for light. When this bends to the left, it immediately creates more shade towards the left and light towards the right. So the next plant in the row bends towards the light, towards the right. Then it creates shade here and light here. Next plant bends in the opposite direction, and so on and so forth. This is a response which involves the perception of the light cues by the plant and a change in growth. It's not just a change in straight growth, as in the case of Arabidopsis. It's differential growth in one side and the other of the stem that causes bending. But it is important. This is a commercial crop, and if we prevent this from happening by physical means, we do not allow the plants to bend, this actually reduces the yield in oil of these crops. So these are responses which have implications for the yield of crops. When we look at the response to warmth, we can see these are plants growing at 23 centigrades, 27 here, but the hypocotyl is also growing much faster. Many have postulated that this has to do with the fact that with the longer hypocotyl, the cotyledons and the more sensitive parts of the plants are far away from the soil, which becomes very hot when there is a strong sunlight. So this might be a reaction to prevent overheating of some organs of the plant. We don't actually think that that's the case. We think that this increased elongation of the stem serves the same purpose as the shade avoidance responses. Why? Because these responses are actually more significant if the plants are shaded. There are a number of reasons, some of which will become evident during my talk. But one of them is that these responses are actually stronger when the plants are shaded. And the fact is that when the plants combine shade with warm temperatures, balance is a disaster. Why? Because shade reduces the amount of CO2 that is fixed by the plant. Elevated temperatures also reduce photosynthesis and increase the expenditure of carbon dioxide by respiration, mitochondrial respiration. So this combination is not very good for the plant, and it requires more light to achieve a balance, in a daily balance in the amount of carbon dioxide. So this reaction would actually be to search for that additional light within the canopy where the plant is shaded. So we think that this response to warm temperatures are actually related to an increment in the shade avoidance response. How is it possible that different environmental cues have such a similar growth responses, at least in this case? Well, the fact is that this is a summary of the sensory perception, the mechanisms of sensory perception of shade and warmth in the plant. And we can see the following. Here we have phytochromes and cryptochromes, which are the known photosensory receptors, which act on, for instance, this is just a simplification because there are many others, but these are two important transcription factors which enhance the expression of growth-related genes. So, for instance, in the presence of shade, the activity of these photoreceptors is reduced, and that allows an increment in the activity of these transcription factors, which actually increase hypocotyl growth. In addition, the photoreceptors act on COP1, which is an E3 ligase. I'm going to talk about this several times during my talk. This is negatively regulated by the activity of the photoreceptors. In turn, this degrades the marks for degradation, some negative regulators of the activity, for instance, of BIB4. So, under shade, there is a reduction in the activity of these, which increases the activity of these, reduces the activity of these, and increases the activity of this. The thing is that when we look at the sensory receptors of temperature, actually 5 years ago, we didn't know any sensory receptor of temperature. Now we know 3. One of them is the same phytochrome B. So phytochrome B is a sensor of light shade, and it's also a sensor of temperature. In addition, there are other 2 known sensors of temperature. One is the messenger RNA of BIB7, which changes its structure under high temperatures, and its translator more efficiently and with this warm temperature structure, and therefore there is more translation, more BIB7, more growth. So, but this is shared by the light shade and temperature pathways. And in addition, this transcription factor, which is part of the circadian clock, is negatively regulated by temperature and also negatively regulates the expression of BIB4. So temperature also affects BIB4 by this pathway in addition to this pathway. And therefore, as you can see here, the effects of shade and temperature converge on the same network. I'm going to repeat some of these steps in subsequent slides, but I simply want you to see here there is a very strong degree of convergence between the effects of shade and warm temperature. Okay, this is part of an experiment where we analyze the transcriptome of plants exposed either to light or shade or to different temperatures in all 4 possible combinations. For instance, light at 20, light at 27, shade at 20, and the combination of shade and warmth, shade and 28. So this is a cluster of genes where expression is enhanced both by 28 compared to 20 and by shade compared to light. So the maximum expression is observed here with the combination of shade and warmth. What are the sort of functions overrepresented within these genes? We can see cell growth, not surprisingly, because the hypocotyl grows more. And we can see genes related to the 3 major growth-related hormones: auxin, gibberellin, and brassinosteroids. Many years ago, decades ago, maybe we could ask, well, how does the environment affect affect growth? Does it affect directly the sort of enzymes that work on the cell wall, loosening the cell wall to increase growth? Well, we know that That is partially true, but the most important action of the sensory receptors of the environment converge on the modification of signaling by these growth hormones, auxin, gibberellin, and brassinosteroids. Therefore, during my talk, I'm going to discuss some of the ways in which either shade or shade and warmth affect the responsivity to, or the signaling degree by auxin, gibberellin, and brassinosteroids. For those who are not familiar with the action of these hormones, here we have auxin, gibberellins, brassinosteroids. These are the receptors. All of them have been identified. These are the receptors of each one of the hormones. At the other end, we have transcription factors involved in the changes in gene expression associated to the action of these hormones. ARFs, PIPs, the same receptors involved in the response to shade and temperature, and BES and BIZAR in the case of brassinosteroids. In between here we have OX-IIAs, which are transcriptional regulators. They actually bind ARFs bound to DNA, and they recruit to DNA negative regulators of transcription, and therefore they reduce the activity of ARFs. DELAs actually physically interact with PIPs, and they bind to PIPs at the same position where they should bind DNA, so they reduce their DNA binding capacity. And BIN2 and other members of the family are kinases which phosphorylate BES1 and BIZAR, reducing their abundance and activity. So let's start with auxin. How does shade affect auxin? For this hormone, I'm going to just refer to shade. Well, under sunlight conditions, there is high activity of the photoreceptors. Phytochrome B, cryptochrome 1 are the most important in Arabidopsis, and therefore these receptors repress the activity of PIPs and repress the activity of this E3 ligase COP1, and therefore the negative regulation. There is little activity of PIPs, and these are further repressed by these negative regulators of their activity. So hypocotyl growth is reduced. Under shade conditions, shade reduces the activity of the photoreceptors, phytochrome B, cryptochrome 1. Therefore, there is an increase in the activity of PIPs. Some of the members of the family increase their abundance and their DNA binding capacity. One of the members actually migrates to the nucleus more intensively in response to reduced activity of phytochrome B. And actually, in the nucleus, this activity builds up, and these transcription factors bind the YACA genes, among others, many others. But these YACA genes are genes corresponding to enzymes involved in the synthesis of auxin. So under shade, there is less activity of the photoreceptors, more PIPs, more YACA gene expression, more auxin, which actually promotes growth. We can see here, for instance, that we refer before to the increase in activity of PIPs. This is the case of P4. We can see that rather rapidly, in response to shade compared to white light to light, in response to shade, we have an increment in the abundance of P4. And that yields an increment in let me show in further detail this other regulatory branch. We can see here that in response to shade, there is a reduction in the activity of the photoreceptors, and COP1 reaccumulates in the nucleus. For instance, we can see here very little COP1, but in response to shade, there is much more COP1 in the nucleus. This increase in COP1 in the nucleus, this is an E3 ligase. E3 ligases actually ubiquitinate their targets, which are therefore degraded in the proteasome. HFR1 is one of the targets of COP1, and we can see here that in the wild type, in response to the time in shade, there is a very important decrease in the abundance of this negative regulator of PIPs. Actually, HFR1 binds PIPs, reducing its binding capacity to YACA. In the COP1 mutant, of course, this effect is much smaller. This is a weak allele of COP1 because it doesn't live long enough. The null allele does not survive long enough, so we have to use a weak allele. But still, you can see very little response of HFR1 to shade in the absence of COP1. We can also see that COP1 in that way affects auxin. We can see here that in response to shade, in the Columbia wild type, there is an increase in auxin in response to shade. That is, of course, lacking in the PIF multiple mutant, but it is also absent in two different alleles of COP1, confirming this actual pathway here. We can see the impact on hypocotyl growth, and we can see the relationship between COP1 and HFR1 in this experiment. In these different boxes, you have here hypocotyl growth, and you have here different genes which are all targets of PIFs. This includes one of the YACA genes, YACA8 here, but there are several others. You can look at any one of these boxes because all of them show the same pattern. You can see here, first, that shade increases the growth and expression of these genes. Second, that this response is absent in the COP1 mutants. And actually, this response is restored in the case where we combine the COP1 mutations with the HFR1 mutation. That means that the function of COP1 is to reduce the activity of these negative regulators to promote growth or gene expression via PIPs. And therefore, if we don't have COP, this pool is very large, repressing the activity of PIPs. But if we don't have COP and we don't have HFR1 either, there is not much of a repressor. We are going to talk about DELA later on. And therefore, the activity of PIPs increases, and we regain the response even in the absence of COP1 because the repressor is also out. Okay? Well, it's true for the first hours of the experiment, because if we look what happens with shade several hours later, we go there and we measure auxin, the difference is already gone. So YACA is no longer having a stronger expression under shade. I mean, for instance, if you go the day after beginning shade, pre-stimulation levels. So hypocotyl growth continues to be fast, but auxin is no longer at high levels. So how is this actually working? Let me remind you of this pathway. We have auxin, the receptor, the negative regulator, OXIA proteins, which recruit negative regulators here to the ARFs, bound to DNA. But let me remind you that ARFs increase the expression of growth-related genes, but also increase the expression of OXIA genes. So there is a loop, regulatory loop here, where where auxin actually reduces OXIAA proteins, but then promotes OXIAA expression. This loop is important to understand what follows. This is the pathway that we propose promotes growth in the long run. If I go back here for a second, ARFs are a family of transcription factors, not just one, there are several of them. OXIAA is a family of negative regulators, not just one, there are many of them. And therefore, we can think about OXIAAs that repress more the ARFs related to growth, others which are repress more the ARFs that repress the expression of, that promote the expression of OXIAA genes. And that is what we think happens here. PIPs, in addition to bind YACA genes, actually bind directly the OXIAA 19 and 29 genes, and they increase their expression. And these two OXIAAs negatively regulate the expression of this IIA 17. Why is that? Because if we go back here, OXIAAs can negatively regulate the expression of other OXIAAs. So these two are positively regulated by PIPs and then negatively regulate this another OXIAA, a different number, which is a strong inhibitor, repressor of growth. And let me show you the data. For instance, we say PIPs. How do we know that PIPs are important? We have measured here the levels of P4 in plants exposed to white light, shade for 4 hours, and shade for 24 hours. In the case of the cotyledons, we can see a very rapid promotion of P4 abundance, but then it goes down. This, the following day, has already gone down in the cotyledons, not to the original level, but at least halfway through. And we think this might have to do with the fact that actually we don't see more auxin. One of the reasons why auxin goes up is because PIPs go up. When they go down, they might explain why auxin is already back to the pre-stimulation levels. Okay, but when we look at the stem tissues, the hypocotyl tissues, in two different tissues, the epidermis and the vascular tissues, we can see here that during the 24-hour measurements, the levels of P4 are at the very least as high as the previous day, or even higher than during the first 4 hours. So P4 is sustainably at high levels with short events of shade and with persistent shade. So this is able to work during prolonged shade. The expression of IIA 19, this one here, increases with shade, but particularly during the second day, consistently with the fact that this is a direct target of PIPs. In addition to being controlled by ARFs, which might be important during the first day, they are direct targets of PIPs. So PIPs, which increase or maintain their abundance during the second day, during the first 24 hours of the shade event, increase the expression of IIA 19 to a larger degree after 24 hours of shade. And that, particularly that extra response observed under prolonged shade, is fully dependent on P4, which is the one we saw to increase during the second day. So these observations support this part of the model. And here we can see this increased color here is an indication of increased activity of the promoter. In this case, we are testing this, the promoter of this gene. This is a promoter of this gene and the control conditions. This is the activity of this gene and the control conditions, and by activating the expression of IAA19, we can see here that when this effector is activated, the activity of the IAA17 promoter is reduced, supporting this negative regulation here. Furthermore, instead of using IAA19 as an effector, we can use in this experiment before as an effector. So going a step backwards, see what happens here if we increase this. This is in Nicotiana pentamiana, so we can see the activity of this promoter is reduced by the expression of before, which is consistent with the fact that before enhances the activity and expression of IAA19, which in turn represses the activity of IAA17. Okay, so auxin is important. Auxin signaling is important. But during the first hour, it has to do with an increase in the hormone itself. Later on, sustained growth of the hypocortical, because the shade avoidance response continues for days and days and days, that response relies on auxin, but is mediated, among other things, but by these changes, not by enhanced auxin levels. In other words, the shade-related signaling here is affecting auxin-related signaling, in this case, downstream the hormone themselves to promote growth. Let's have a look to the second hormone of this series, gibberellin. This is the pathway of action of gibberellins. We saw it before. This is the receptor, and these are the DELA proteins which inhibit PIPs. So the canonical way of degradation of DELA proteins is via the GID receptor in response to changing in the hormone gibberellin. Here we show that there is also a direct action of the pathway involved in the response to shade, mediated by COP1 acting directly on DELA proteins. Under shade, there is a reduction in the light input, there is a reduction in the activity of the photoreceptors, and therefore there is an increase in the nuclear accumulation of COP1, and COP1, we propose here, directly degrades DELA protein. I mean, these experiments were quite difficult because in terms of hours, shade or warm temperature actually increase the levels of gibberellin. So here we have to demonstrate that the effects of shade and warm temperatures can be the direct result of COP1 activity, and in physiological experiments where we have kinetics analyzed to investigate precisely the kinetics of this response, we have seen that the effect of COP1 was very rapid in the terms of 10 minutes could reduce the abundance of DELA. But the question is that because COP1 somehow is affecting GA levels, the levels of gibberellin? Or it is because COP1 is directly acting on gibberellins, inducing their degradation. Because also GID1 induces the degradation of DELA. Well, to address that, we use a mutant form of one of the DELAs. RGA is one of the DELAs. There are 5 in Arabidopsis. RGA is one of the DELAs. We use the mutant form of the DELA proteins, which is not recognized by the GID1 promoter. So the DELA is still there. It is still able to act on PIPs, but it is just mutated in a domain which is recognized by GID1. We can see here, for instance, we add a lot of GA to this RGA. We add a lot of gibberellin, and nothing happens. Why? Well, because you increase gibberellins, you activate GID1, but GID1 is unable to recognize this particular mutant form of RGA. But yet, we can see here that these are confocal experiments. We can see in green the fluorescence of this DELA protein RGA, mutant form, which is unaffected by gibberellins. We can see that although they are not affected by gibberellins, they are still reduced by warm temperature. They are still reduced by shade. And actually, if we look at this, this is the same experiment as this one, but now instead of doing it, instead of having this in the wild-type background, they are in the COP1 mutant background. And we can see no degradation. Therefore, shade or warmth can reduce the abundance of RGA in a mode that is independent, it does not directly require gibberellin, but requires COP1. In further experiments, we found that there is a direct physical interaction between the COP1 SPA complex and DELA proteins. And we also observed that COP1 ubiquitinates DELA proteins such as RGA in vitro. So this pathway is confirmed, and it actually affects the growth response. So you can see here a response also shared by shade and warmth. We can see here, for instance, the kinetics of the normal RGA. This is not the mutant. We can see that RGA decreases in the wild type, decreases in response to shade. In the wild type, it decreases in response to warm temperatures, but that decrease is much weaker in the absence of COP1. Simultaneously, we can see here that COP1 in the nucleus increases with shade and increases with warm temperatures, supporting the model shown here, where shade increases nuclear COP or warm temperatures increase the nuclear abundance of COP1 and therefore decrease the abundance of DELA proteins in a COP1 dependent manner. So the conclusion here is that, again, environmental signals such as shade or warmth affect signaling downstream growth hormone, in this case gibberellin, affects signaling downstream hormone to promote the growth of the hypocotyl. The final slides are about brassinosteroids. And here we are going to shift to, I'm going to show you some hypocotyl results, but mainly we are going to shift to a different response, which is the growth of the cotyledons. We can see here in this slide here that whereas, as I showed you before, the hypocotyl grows more in response to shade or warm temperatures, we can see here that the cotyledons, which are the embryonic leaves of the seedling, the cotyledon expands less under shade or warm temperatures. So the other way around. So our first question here was, okay, are any of these hormones involved in this growth response of the cotyledons? We know from some experiments I show you and others that have already been published that all three growth hormones are important for the hypocotyl growth response. Okay, but which one of them is important for this? Let's have a look. Here we have the measurement of RGA, the one that dropped in response to shade and warmth into hypocotyl. What happens now in the cotyledon? Nothing. So we don't think that gibberellin has to do with this drop in the growth of the cotyledon. What happens with DR5? I showed you some DR5 data before. That was with gas. This is another reporter, but it's the same promoter. As expected, in response to shade or response to warmth that activate PIFs and YACA genes, there is an increase in the activity of this auxin reporter under shade, under warmth. However, when we look at this mutant, SUB3 mutant, is a mutant partially affected in the synthesis of auxin. What happens here is that when there is less auxin, the cotyledons grow less, no more. Therefore, this increment in auxin levels does not look to be the cause of this reduction in cotyledon expansion. Why? Because if we reduce auxin levels, we actually see a reduction and not an increase in the growth of the cotyledons. However, here we measure BES1. BES1 is one of the main transcription factors of brassinosteroids. And we can see here that there is an important reduction in the cotyledon in response to shade, in response to warmth. This is a mutant deficient in the synthesis of brassinosteroids. What happens here? Well, you can see here that there is a reduction in the expansion of the cotyledons and actually a lack of response to shade and warmth. In this case, okay, this is consistent with the possibility that a reduction in the activity of brassinosteroid signaling affects the growth of the cotyledons in response to shade and warmth. Why? Because this is an indication that brassinosteroid signaling is being decreased, and this is an indication that when brassinosteroid signaling is decreased, you actually have a reduction in the growth of the cotyledons. Okay, here again we have cotyledons, abundance of BES1, reduction with shade, reduction with warmth. Here we have the other transcription factor. Remember, they were BES1 and BZAR1, the two main transcription factors involved in brassinosteroid responses. It also decreases in the cotyledon with shade and with warmth. This is very interesting here. You can see here that when we look with a confocal microscopy in the hypocotyl, BES1 goes up with shade, BES goes up with warmth. Same holds true for BZAR1. Therefore, this is a growth promoting hormone. There is a correlation between the growth of the cotyledon and the decreased growth of the cotyledon, the decreased levels of BES1 and BZAR1, the increased growth of the hypocotyl, and the increased nuclear abundance of BES1 and BZAR1, which actually promote growth. Couple of questions here. First, we did Western blots. These are Western blots with not with Arabidopsis thaliana because it's too small to separate the cotyledons and the hypocotyl. We used a brassica, very related to Arabidopsis, but a bit larger. We can see here in Western blots, quantified here, that the phosphorylated and the dephosphorylated forms of BES1 are reduced in the cotyledons in response to shade or warmth, reduced in the cotyledons in response to shade or warmth, and increase in the hypocotyl in response to shade and warmth, increase in the hypocotyl in response to shade and warmth. Therefore, these changes in nuclear abundance are, at least to some degree, due to changes in the abundance of the protein. One of the possible explanations for this might be, okay, brassinosteroids, the hormone, the chemical signal, is actually decreasing in the cotyledons and increasing in the hypocotyl. That's a reasonable hypothesis, because that would explain the changes in BES and BISAR by the canonical pathway. However, we have measured this, I didn't put here collaborators, but never mind. And we can see that there is no decrease with shade and warmth, no decrease. Actually, there is some increase in some cases. There is no decrease in the cotyledon, which might explain the decrease in BES1 and BISAR1, and there is no increase in the hypocotyl that could fully explain the observed increase of BES1 and BISAR1. Therefore, it's not the hormone itself which is explaining this, there must be something else. And that something else might come from some of the components of the shade signaling response. So we tested, for instance, what happens with phytochrome B, COP1, and PIF4. We did first these experiments to see that in the cotyledons, these three components change as they do in the hypocotyl, and that is true. I mean, we can see the results here. But let's concentrate here. We can see that this is a wild type, this is the 5B mutant, this is the wild type in cotyledon hypocotyl, this is the 5B mutant. So 5B actually enhances the abundance of BES1 in the cotyledon and the hypocotyl. Let's have a look here. COP1. In the wild type, there is a drop in BES1 in response to shade, in response to warmth. In the COP1 mutant, that response is absent. I mean, this decrease requires COP1. Remember, COP1 is an E3 ligase. In the hypocotyl, the effect of COP1 is completely different. And if I had time, I'd give you a word about this. Finally, the PIF4 mutant. There is a decrease here. In the PIF4 mutant, there is no decrease. So somehow these components could be affecting the abundance, or are affecting the abundance of BES1 in the nucleus. But how? Well, PIF4 is a transcription factor. It could be affecting the expression of BES1, and it does. Here is the expression in the cotyledon, which is reduced by shade and warmth. And the levels of expression, there's still a response here, but the levels of expression are higher in the PIF4 mutant, indicating that this is actually a repressor. of the expression of BES1. These two also affect the expression of BES1, but in ways that suggest that they are acting through BIB4. In addition, we can see here that BIB4 actually binds different regions of the promoter of BES1. These are experiments under conditions with high red, far red, which is no shade, all shade conditions. There is no significant difference between shade or no shade, but in all cases there is an interaction. And that lack of difference in the response to shade may be due to the fact that this is already an overexpressor of P4 that we use for these experiments. Okay, P4 would affect BES1, as we saw in the last slide, because it affects its expression. So I think I have another 5 minutes or so. Okay. Well, what happens in the so we question the following. Okay, can we explain everything in terms of BES1 expression? Is all the regulation related to the fact that P4 affects BES1 expression, or there is something else? So we did the following. We analyzed, we had a number of plenty of data here in terms of levels of BES1, and we had levels of expression here for the same genotype. So we question, okay, can we explain these levels in the cotyledon? Can we explain these levels of abundance, nuclear abundance of the protein, by these levels of expression? So we did multiple regression analysis. We can see here that, well, as expected, the expression of BES1 has a significant effect, positive effect, more expression of BES1. We expect more abundance of the BES1 protein. This here, more expression of BES1, more abundance of the BES1 protein. But in addition, to explain this data, we need an additional action. It's a little thing about phytochrome B, but mainly a significant effect of COP1 to explain the protein after we have considered the level of expression. Of course, P4 affects the abundance of BES1, but according to this analysis, all the effect of P4 can be accounted by the fact that the P4 affects the expression of BES1. So we can see here, for instance, two things. First, if we use an inhibitor of transcription, there is less BES1, but there is a response still. So something else is working, and that something else is no longer working in the COP1 mutant. So this could be related to the action of COP1 as an E3 ligase, inducing the degradation of BES1 in the proteasome. But if that is true, we used an inhibitor MG132, which inhibits the degradation in the proteasome. What we can see here, for instance, for the cotyledons is, okay, under shade of warmth, there is a reduction in BES1, but that doesn't occur in response to the application of this inhibitor of proteasome degradation. So this has to do, at least in part, with degradation in the proteasome. And here we can see that, for instance, in the COP1 mutant background, we don't see any effect of the inhibitor of the degradation in the proteasome. What is the logic of this? Well, this is actually suggesting that COP1 induces the degradation of BES1 in the proteasome. So you can cut this pathway either by eliminating COP1 in the mutant or by using the proteasome inhibitor. But you cannot kill it twice. I mean, once you lack COP1, MG132 has nowhere to act. Okay, you don't see an effect here. Conversely, you can see here in the P4 mutant background, there is more BES1. Remember, it increases the expression of BES1. And in addition, you can increase the abundance even further by increasing, by reducing the degradation of the proteasome. Why? Because P4 is acting on expression, and later on, by eliminating this branch here, we increase the stability of the protein synthesized from that extra expression of BES1. Okay, this is, I will skip this. This is just to show that there is a direct interaction between COP1 and BES1, physical interaction tested by different means. And finally, to show you that actually, BES1 promotes cotyledon expansion and hypoxyl growth. I mean, a genetic proof that those changes in the dynamics have anything to do with the growth of the cotyledon. You can see here in these pictures, the wild type has larger cotyledons than the BES1 mutant. We can see here the quantified data. In the BES1 mutant, the cotyledon expansion is already low in the under control conditions and no further reduced by shade or warmth, which is consistent with the observations I presented before. And with that, a summary of our model of control: shade and warmth reduce, among other things, they reduce the activity of phytochrome B. Phytochrome B, reduced phi B actually allows P4 and other PIPs to build up their activity, and COP1 is also, that's also true for COP1 in the nucleus. We have more expression of BES1, and we have more activity of more stability of BES1. In the case of shade and warmth, when P4 increases, this is repressed. In the case of shade, phi B is reduced. This is increased. This is reduced. So we see a reduction here, here, and here, which actually reduces expansion of the cotyledon. And the final test of that is, if we measure the expansion of the cotyledons, we can predict that if this is true, if I use inhibitor of the activity of COP1, I can actually reduce the impact of shade and warmth, and that is actually the case. And that occurs in the wild type. In the COP1 mutant, it actually has no differential effect. The application of this MG132, we have high expansion and high expansion, irrespective of application of the inhibitor of the proteasome. You cannot act twice on the same pathways that are already no longer working. So what are the conclusions? We have 3 growth hormones here. Are they necessary for growth responses? Yes. There are many examples in the literature. The mutants do not respond well. Other changes in hormone levels? Well, yes, for auxin, very rapid. Gibberellin, yes, but because there is a change, but these changes take hours, and the growth responses are much faster. And in the case of prasunesteride, no, not at least in the time framework we look at. In all three cases, the signaling and perception mechanisms of the hormone are affected in response to shade and warmth. And with that, I would like to show you the people that work with me, and this is our current composition of the group, and thank you very much for your attention.
Thank you very much, Jorge, for the wonderful talk. And is there any question? I cannot see anyone here in the Meet chat. What about you there, Leandro?
Can I start? I have a couple of questions.
Sure.
To Jorge.
Sure.
Jorge, thank you for your wonderful presentation. Very interesting results. I was wondering, as you show that warmth and shade affect brassinosteroids signaling pathway, those environmental conditions, do they affect, or did you also observe any abnormalities on the vascular development? Because brassinosteroids are also known to affect vascular development, and also BIS1 is a promoter of xylem differentiation. So my first question is whether did you notice that these seedlings grown under warmth or shade, if they have an abnormal vasculature development as well?
Mm-hmm. Well, there are some data in the literature. We have many years ago analyzed a cucumber mutant which is deficient in phytochrome B, which is very long, as if it were under shade all the time, or warm conditions all the time. And it had a reduced development of the xylem and other stem tissues, so, but particularly the xylem. So it might be.
Okay. And another question that I have, I mean, you knew, I think that you chose BIS1 to analyze because you knew that is one central transcription factor in the signaling pathway of brassinosteroid. Did you analyze, or did you know if this pathway also affects an upstream step, which is JSK3 kinases? These kinases, they inhibit BIS1, but they are also involved in the inhibition or controlling auxin pathways and other developmental pathways. So did you try, or do you know if you have any evidence that your system or these conditions are also affecting the SK3?
Yes, I mean, there is a paper, we haven't analyzed that in these experiments because we focused on these on the pathways I described, but there is a paper, a current biology paper describing the action of shade on some of the phosphatases that in turn affect the kinases. So something is known. It's a paper that talks about shade and effect of salt. If you want to search for it, it's a current biology paper. That's one of the things I remember from the literature.
Okay, thank you very much.
Anything there in the YouTube, Leandro?
Not yet.
Okay, so I have a couple of questions. One actually is, could you tell me something else? You mentioned that hypocotyl elongation in response to high temperature, it's actually a way to increase photosynthetic activity to deal with the increment in respiration rather than a question of temperature itself. So which are the evidences regarding this affirmation, this sentence, this statement?
Okay. We have a paper which has just appeared in New Phytologist, which is dealing exactly with this hypothesis. I summarize very briefly the evidence. First, we have done a side-by-side analysis of the quantitative impact of a number of mutations that affect the response to shade. So the question there is the impact of the mutations different or similar in one case and the other? And all the mutations but one, which was ELF3, affected quantitatively the same extent the response to shade and the response to warmth. And we analyzed different accessions of Arabidopsis thaliana, different genetic materials of Arabidopsis thaliana, and we measured the response to shade and the response to warmth. And the correlation between the response to shade and the response to warmth was huge. And that wasn't related to the background growth rate. What's the meaning of this? The meaning is that there is a very strong genetic linkage between the response to shade and the response to warmth. They actually serve to very different purposes. One is to shade and the other is to temperature. Why should they be genetically linked? I would expect them to be easily isolated one from the other, because a plant may have shade and no warmth. So you want tools to touch one, not touching the other. So that's one evidence. Another thing is, for instance, we look at the patterns of response to temperature. The response starts in Arabidopsis at 10 centigrades, and there's a linear growth response, for instance, between 10 and 30. So what we did was to analyze temperature patterns in different parts of the world, and we said, okay, let's say 35 is a heat stress. Okay, so we analyzed the probability of having a heat stress as a function of the mean temperature the previous 4 days or something like that. If you have a temperature of about 10, 15, and so on centigrades, the probability of having a high temperature the days after is practically zero. The probability begins to grow up slowly when we go up above 20. So why is the growth response increasing from 10 onwards when there is no risk of having a heat shock, if the probability of heat shock begins at a much higher temperature? It doesn't make sense. Why is it that we compare the growth to the response to temperature in a plant which is in the light and the plant which is under shade? Which responds more? The plant which is under shade. So I would say, well, why would shade reduce the response to warmth if that is related to a heat shock? We all know when we go to the beach, the probability of getting a heat shock is not under the umbrella, but under the sun. So there should be a stronger response to avoid heating under the sun, not under the shade. So there are a number of pieces of evidence that work against, at least in Arabidopsis. I mean, there are other things in other species, okay? The other day, someone sent me a reprint with results in grape, where warmth increases the density of stomata. And that could indeed be a response to reduce the probability of heat shock.
The
okay?
temperature, yeah, for sure.
Yeah.
Exactly. And another detail that maybe I missed, but still talking about hypocotic elongation, when you talk about DELA role in that signaling pathway, have you tested that in gibberellin mutant, biosynthetic mutants? So it's that DELA role is independent of Giverelli.
Yes,
acid.
well, we did not test, for instance, you mean if we used, we introduced the DELA reporter, for instance, into a gibberellin synthesis mutant, okay?
Exactly.
Well, we didn't do that. We used inhibitors of gibberellin synthesis. And we built up a response curve to the inhibition of gibberellin, and we used a saturating level of the inhibitor of gibberellin, paclobutrazol, and still we have a response to shade and warmth, which is absent in the GOP1 mutant. So it's not genetically, but pharmacologically more or less the same. Great,
exactly. Yes, and my last question is actually more a philosophical question. So according to the scenario that you have shown, there's no way to uncoupling temperature and light perception.
I mean, it's likely that we have not seen all the genes first, but I would say that they are not very easy to uncouple. But when we look at the growth response of the hypocotyl, if we talk about, for instance, other temperature responses, they are another world. I mean, for instance, if we control about the flowering by temperature or vernalization, they are completely independent from this.
Yeah, sure.
That's another story.
Great. Thank you very, very much. Any other question?
I have one. What is I know that your goal is about the shade effect in this plant, but what is expected under the dark conditions, because there's no light and then the temperature is low?
Okay,
about this expression signaling.
Yeah. Okay, well, that's a very interesting question.
Sorry. Is there a residual effect about this? No.
Yes, I mean, we are talking about, we have working in relation to darkness, but the darkness of the night, for instance. Mm-hmm. And we have experiments that show, we did two things. We analyzed whether the effect of daytime temperature affects nighttime growth, and whether the effect of nighttime temperature affects daytime growth. And both are, the response to both is actually yes. So for instance, growth during the night depends on temperature during the night, mm-hmm. but also on temperature during the day. And we are finding the signaling components that carry the response from one part of the cycle to the other, okay? In the case, for instance, the status of the temperature during the night affects phytochrome B, not the sensory activity of phytochrome B, but the abundance of phytochrome B, and that affects what happens during the day. It's, I mean, it's a set of things in their own right, but they affect the abundance of phytochrome B, and that affects the phytochrome B left for the subsequent day, for instance, and affect growth there. And temperature during the day affects, of course, P4 levels, and there is sort of hysteric control of phytochrome of P4 levels. We are doing the experiments nowadays, but the thing is, for instance, if the expression of P4 goes up during the day and you drop the temperature during the night, P4 expression keeps high. So it doesn't respond very well. It goes up, but doesn't go down very easily. So there is a sort of a hysterical behavior. It doesn't go in the same way when it goes up, the temperature, and goes down. So that involves a carryover of daytime temperatures into the night.
Mm-hmm.
So, yes.
Great. Very complex.
Yes. Thank you.
Great. Anything else?
I have one last question. I mean, also kind of not philosophical, but I mean, Jorge, you started your talk with this picture of a soybean field. But then all the work that you reported was done in Arabidopsis, which is, let's say, just a model plant. Do you have an intention in your lab? I'm not sure if you use really crops, or do you envisage how this knowledge on this basic science, on how the plants respond to the shade and to temperature, how this could be applied to biotechnology of crops, how to make, for instance, more resilient plants in this changing environment that we are living in?
Yeah, for instance, of course, we do most of the basic experiments, or all the basic experiments in Arabidopsis, but then we try to see how that can be somehow translated to crop species. And for instance, well, I show you some results from the past with sunflower. We are currently doing experiments with maize, and with all this information about the effects of temperature and light on Arabidopsis, we built up a model for hypocultural growth, and we are seeing how we can predict the growth of maize using that Arabidopsis model. Of course, the numbers are different, but we want to see whether we can apply the principles. And actually, I mean, we still have many things that require rethinking or whatever, but we did something very simple. We said, okay, how much does a maize plant growth and the optimal conditions? And we use the Arabidopsis model, just as it was, to reduce that growth from the optimum. And I mean, it works reasonably well. I mean, there is something there. And so another interesting thing is that we have analyzed the physiology of maize plants, how that physiology has changed with the eras. I mean, we have old maize cultivars and new maize cultivars, all a series of maize cultivars. What are the changes between? Why do they yield more?
You mean how, for example, how the domestication affects temperature and light perception? Yeah,
I mean, how breeding during the last century affected that. I mean, it's already known that one of the changes is that the fact that current maize varieties are able to grow at higher densities. So the commercial companies use higher density, but for that they had to get the genotypes that are able to grow close one to the other without losing yield. And we analyzed the changes, and one of the things we found is that modern varieties actually have more photosynthesis. They manage to get more photosynthesis of the leaf that supports the ear. I mean, that gives yield to the corn plant. Okay, so it has to penetrate more light and have more photosynthesis. We measure photosynthesis, and modern cultivars have more photosynthesis, and in these particular leaves, not the whole canopy, this particular leaf. And they also have a reduction in the respiration of these leaves. So they manage to support higher densities, more shade, by reducing respiration of the leaf that has to support. The leaves are thinner. This has to support the growth of the ear and the yield. And this is interesting because respiration is a parameter strongly affected by temperature. So it seems that by getting
reinforce the answer you gave me to my first question. Amazing, great.
Magda, we have one question at YouTube. Do you want me to read for
Sure.
Jorge? Okay, so Jorge, Daniele Rosado said that your talk was great, and she asks you, how can the temperature at night affect the levels of Phi-B during the day? Is the protein degraded or the transcript affected? She said that she does understand that during the day, high temperature leads to inactivation of Phi-B, but she cannot understand the relevance of night temperature in this case.
Okay. Thanks for the question and thanks for the words. The thing is, what is the basic sensing temperature of phytochrome B? It has two interconvertible forms. One is inactive and the other is active. So light absorbed by this form generates the active form. Light absorbed by this form generates the inactive form. So this changes with shade, okay? That is shade. Light absorbed by one form or the other, phototransformation, and active or inactive form. In addition, there is a reaction that goes from the active form to the inactive form. And this reaction depends on temperature. It's faster, there's a faster inactivation when temperature is warm. So that is the basic sensor activity of phytochrome B. But now, there are consequences of that. For instance, during the night, it depends, temperature can affect this reaction, but as a result of that, these two forms actually have different turnover rates, okay? The inactive form is more stable as a protein, it's more stable than the active form. So eventually, if you have more reversion to the inactive to the active form, you are storing phytochrome in a form that is more stable. So that happens in the dark, but next day that inactive form, when it receives light, it becomes active. So you saved in the bank the phytochrome for the next day. Okay, so it's a way in which it's not a basic sensory function of phytochrome, but affecting other aspects of the phytochrome dynamics. It's how it is affected. Okay, how nighttime temperature can affect daytime abundance of phytochrome B.
Perfect. Thank you. Daniele Rosado has also another question based on your explanation about the maize varieties. She's asking whether these maize varieties have different leaf angles or plant architecture compared to other varieties that would help increase light capture.
Well, there are indications from the work of other labs that there are some changes in architecture. For instance, we actually measured more the amount of the area of the leaves actually is more or less the same. It's not a very obvious trend. I mean, in some countries, because they have different varieties, you can see small trends, but it's not very large thing. But it's more or less general that the leaves, the upper leaves are a bit more upright, so they allow more penetration of the light. Well, we can see that there is more light at that level. Okay, there is more light there, reaching there. That is probably because of that angle, which then changes as we approach the base of the canopy, and the leaves are able to intercept. We have measured that leaves are actually able to intercept that and use that for photosynthesis. And in addition, there is a reduction in respiration. But yes, there is a change in the architecture. We didn't measure that directly, but the stronger penetration implies that.
Okay, thank you. There's no more questions at YouTube, only compliments about your great talk. So,
thank you very much.
Well, Jorge, it was a wonderful class. We thank you very, very much. And it was a pleasure having you here. And if there's no more questions or comments, I just say you goodbye to everybody. Take care, and we keep in touch. Thank you very, very much.
Thank you, Magda. Thank you all for your attention. And bye.
Thank you, Jorge.
See you then.
Bye-bye.
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 Bye.